It is the Dairy Standard Agency's second webinar for this year, and we are really so glad that you could join us. I'm Jacqueline from SA, and with me I have Jan Klopper from Plaas Media, who will facilitate the question and answers at the end of the presentations. Special thanks to Plaas Media for the marketing mentioned. We have a question and answer session in the presentation, so you are welcome to either write your question in the question bar. At the top there's a question bar, or you can raise your hand during the Q&A session, and then you can ask your question directly to the presenters. So either way, please make sure that you give us all your questions. Our topic for today is a really relevant, crucial topic, and I'm going to give it. It is a cost-effective approach to contaminant reduction in dairy effluent. And we are really in the fortunate position to have 4 experts with us to share their knowledge and their expertise, their experience. And it gives me great pleasure to present and introduce our 4 speakers for today. First, we have John McGosh, environmental scientist with over 20 years at the Institute of Natural Resources. We have Dr. Nkomisa Mpongwana. I'm going to name all the names, Dr. Faiz Ansari and Dr. Lois Kuba. All 3 of them are postdoctoral researchers at the Institute for Water and Wastewater Technology at the Durban University of Technology, DUT. Due to a time constraint, I'm not going to read their biographies, but you've seen it when you registered for the webinar, so you know the expertise we have in this webinar room today. We are really fortunate to have you. But without further ado, over to you, John, and we are looking forward to your presentation.
Thanks, Jacqueline, and thanks everyone for taking time out of your busy schedules to join this webinar. Yeah, I think, you know, as Jacqueline says, it is very important and very pertinent, and I know that sort of the dairy standard focuses on compliance in terms of the quality of the milk being produced, but I think as responsible producers, it's also necessary to consider minimizing and mitigating potential negative impacts on the natural environment as well. And this has been a project that we have been involved in over the last 3 years with the Durban University of Technology and the Institute of Water and Wastewater Technology around investigating wastewater in the dairy sector. And before I go into the presentation, I'd just like to acknowledge the support received from Milk South Africa in contributing to the completion of this research, as well as the Joint Nature Conservation Committee based in the United Kingdom, who have also supported this research. So to start with just a bit of background and context, the Institute of Water and Wastewater Technology, as the name indicates, is leading various innovations around water and waste management, one of which is pica remediation, which we'll explore in a bit more detail shortly. The INR is a Pietermaritzburg-based organization, a non-profit company, whose mandate is to support the wise use of natural resources for the benefit of environment and society. But we see ourselves as an organization that takes science to society, taking pure and blue sky research and applying it in real world contexts to see if it works. And yeah, we've been conducting this research project over the last 3 years, so we're quite excited to share some of the results of the work. I think we all know that the dairy sector in South Africa is important. It's one of the largest milk producers in Africa, producing 4.5 billion litres every year. It's the 4th largest agricultural sector in South Africa. So it's very important in terms of the economy, employment, and what, production in South Africa. But at the same time, there are substantial volumes of wastewater generated through primary production and through the processing sectors, varying from 3 to 11 million meters cubed per annum. And a key component, particularly at the farm scale, is lots of nitrates, ammonium in particular, phosphates, suspended solids. There's also potentially a wide range of pathogens from animals that are perhaps infected or sick. At the same time, there's high organic content on farms and associated very high chemical oxygen demand and biological oxygen demand. And these can impact negatively in terms of making anaerobic conditions within water sources, and also impact potentially on soil health and soil productivity if applied to soil in the long term, and also as clean in place chemicals as a result of the washing that occurs within the farms. So yeah, I think it's important to consider these aspects and how do we manage them effectively. Currently, our research suggests that most wastewater systems on farm are a simple slurry pond which stores water for irrigation, and the pond is, when it reaches capacity, it's drained through irrigation systems, and then possibly on an annual or multi-annual basis, it's dredged, and that waste slurry is applied onto fields as well. So if it's not managed correctly, there is a severe risk of contamination, both of soil and water bodies. Certainly in KZN, you would have known that we've had a very wet summer, and with a lot of surface runoff, this can potentially collect into water bodies and impact negatively both on ground and surface water sources. And finally, there's a growing focus on environmental stewardship, being better environmental managers. I'm looking at regenerative agricultural practices, which is receiving a lot of attention locally and international. Issues related to soil carbon, increasing adoption of conservation agriculture and other regenerative practices, better management of fertilizers to reduce inputs, not only to reduce negative environmental impacts, but also for very valid economic reasons due to the costs of fertilizers. So there are sort of efficiency and economic and environmental benefits associated with some of these practices. And I think it's important to see how on-farm and downstream impacts associated with wastewater can be mitigated. And certainly many of us who are living in urban areas and rural areas are feeling the impacts of water quality issues, access to water of sufficient quality and quantity, and finally, it's important for legislative compliance. So to get on to our study, really we have three broad objectives: is to try and understand the wastewater systems and practices that are applied using 4 dairy farms and 2 small-scale processors located in KwaZulu-Natal. We identified them through a process of engagement and ultimately secured support to conduct study on these particular farms. We evaluated the physicochemical characteristics of the wastewater that is generated, and also wanted to see if there was any seasonal variation in the characteristics of the wastewater. And then based on this, investigate the feasibility of green algae or phycoremediation, which I guess is the crux of this presentation, as a potential green solution to treat dairy farm and dairy processor wastewater. And this was done both in lab and also in the field in situ. I'm now going to hand over to our DUT colleagues to just speak a little bit more about what phycoremediation is. Thank you.
Thank you, John. Thank you, everyone for joining this morning. My name is Dr. Faiz Ahmed Ansari. So what is phycoremediation? Phycoremediation is there is a different technology to treat the wastewater. Various wastewater is the domestic wastewater, industrial wastewater, or dairy wastewater. If using algae to treat wastewater is called phycoremediation. So the question arises why there is many technology available in the world, in a market, and locally and globally. Why you are going to use a phycoremediation? So there is many advantage over the other technology till now we are using. This phycoremediation, what it does, you use algae to grow in a wastewater, in a dairy wastewater. What about the nutrient inside the dairy wastewater available? Those are required to grow algae. And when you are growing algae is used CO2. Where what about the CO2 in environment? Nowadays, everyone is talking about the global challenge of CO2, CO2 credit, carbon sequestration. So when you're growing algae in a dairy wastewater, is captured the CO2 from the environment, took the sunlight from the sun, and grow. During growth, what about the nutrient in the dairy wastewater? They used for the metabolic process and produce a useful biomass. And that biomass can be used for different application. In the term of the what's the chemical composition in the biomass, either lipid, oil, protein, pigment, carbohydrate. If there is oil is a more, if the oil is more, we can go for the biofuel production. If the carbohydrate is more, you can go for the bioethanol, biohydrogen. And if they are more pigment, you can go for the high value product. And if the high protein contents, you go for the feed and food applications. So after harvesting, what about the water left? You can use as a different application as a reclaim water. So this is the advantage using phycoremediation. This technology, when you are using, there is no secondary byproducts. Is produced zero waste. This concept is using a phycoremediation for the dairy wastewater treatment. Is a zero waste. So next slide. Next slide. This is the continuous of the previous slides. And when you grow algae in a wastewater for a treatment purpose, there is many advances as a wastewater treatment, CO2 reduction, biorefining concept, whatever the produce the biomass can use for the different application. And this is algae is a photosynthetic organism. When you grow algae and during the treatment process produce oxygen and also capture the CO2 and nutrient took from the dairy wastewaters. As we know, CO2, as in earlier I talked about the CO2. So when you grow algae, if you're growing algae in a wastewater, is capture the all the nutrients for producing the biomass and treat the wastewater. And for 1 ton of biomass, is capture almost 2 ton of the CO2 from the environment. This is one of the advances on the global. People are talking about the global warming and CO2 sequestration and carbon credit. And this biomass can be used for the further application in different feed, food, depend upon the biochemical composition. Algae can be cultivated in various types of system, including open pond, photobioreactor, offer flexibility in design and implementation depending on the land availability and a specific treatment goal. Next slide. Yeah, this is a system, algae cultivation is contained in both the upstream and downstream processing. The first, you can see the microalgae cultivation is contained algae strain selection, nutrient optimization for if we are growing in a dairy wastewater, particularly for phycoremediation purposes. If there is a less nutrient, you need to add more nutrients. You have to see what's the nutrients is not available in your growth media in a wastewater. You have to add, because algae is a photosynthetic organism. For growth, you need all nutrients for the growth to remove the other nutrients. So this process, the first one is called upstream. After microalgae cultivation, all the process like a biomass harvesting to tea application come in a downstream processing, and biomass harvesting after like a 21 days, 25 days, depend upon the where you are growing. Then you have to harvest the biomass for further application. And after harvesting, during harvesting, you can use a different technology. There's a centrifuge, decanter, gravity settling, and the other technology also available. And After harvesting the thick algal biomass obtained, you need to dry for the further application. And drying techniques, you can use sun drying, oven drying, freeze drying. It's depend upon the application and what application you are going for. And if you want to extract any metabolite, like a protein for different application, carbon source for, you need to break the cell to easily extract the metabolite. And further, you can go for the application purposes, depend upon the biochemical composition. Either go for the fertilizer, go for the feed food, or go for the any green energy purposes. Next slide. Next slide, please. Yeah, Dr. Anukumi-san.
Oh, so can you go to next slide, John? So, okay, we did the monitoring of the dairy farms, which we work with, and we work with 4 dairy farms. We monitor the amount of water they produce per day and also the volume which they produce, because when you want to adapt a treatment process, you need to understand how much they produce and how much time your treatment process is going to require. So we find that both for dairy farm, they were producing the amount of wastewater between 30,000 and 60,000 per liter, but it also depended on the amount, on the number of cows they have, and we also measured the slurry pond which they use. You see for farm 1, they had like 2,200 cubic meters, and then going to 3,500 cubic meter of the slurry pond they use. This allows us, when we're doing designing, it helps us to know how much we need, how much pond, how big the pond of the algae we will need. So we did that monitoring before we started. Next slide, before we started with the experiment. And then we did some monitoring of the wastewater, where we took sample every month for about 6 months, and we monitor the contaminant, your chemical oxygen demand, your ammonia, your phosphate. And we find, if you can see the red line is the permitted amount. And you can see that the bars all go beyond the red line, which means the wastewater that is produced, that we took from the slurry ponds, exceeded the concentration which is permitted for irrigation. Next slide.
Thanks. If I can just add, so just for context, the National Water Act, Act 36 of 98, provides various thresholds for irrigation with wastewater that's either domestic or contains biodegradable material. And this includes irrigation with dairy wastewater slurry. And yes, in many cases, we're finding that those thresholds are being exceeded, which I think is just highlighting the need for additional measures to mitigate the quality of the wastewater that is being generated. Thanks. Sorry to interrupt. You can proceed.
Okay. So before we did just a little bit of background, before we did this trial, this experiment, we look into 2 different processes, biological processes that we found that we can use to treat this wastewater. We look into the and we also look into phycoremediation. And phycoremediation emerged as the best treatment methodology, not only because it was superior in terms of treating contaminants, also because of the benefit that comes with the biomass that is found. So when we adopt phycoremediation, it does not require additional step at the end for treatment, unlike the activated sludge would require additional step. You can use the biomass as a biofertilizer, which is, I think it would serve the same purpose that the wastewater, when you're irrigating with the wastewater, it usually serves. So,
except the fact that now the water does not contain high amount of contaminant. So we ended up opting to adopt a FICO remediation for lab trials. And the results that we found, can we go to the next slide, please, John? The results that we found, because the water from the slurry ponds, as you've seen in the pictures, is kind of like turbidity, and it's a very dark brown, sometimes black. And you will recall that my colleague here have mentioned that algae work by it's a photosynthetic organism which work by absorbing the sun for energy, and also then that's how it grow. It absorb the sun. It absorb the light from the sun. So when the water is in that color, it sort of like interrupt the penetration of the sun into your algae pond. So we had to dilute the water. So how we diluted the water? We diluted the water by adding 30% of water, and then we get we added 70%, and then we get 30% of dairy wastewater, and then we added also 40%. We just wanted to see at which dilution we will get highest removal efficiency, and then the other experiment, we didn't dilute them. And we find that when we dilute with 40% fresh water, and also we dilute with 70%, particularly for the dairy farms, we find that for CODs, we get higher removal of CODs at 30% dairy wastewater and 60%. The main reason is because when you're making the dilution is because you're reducing the turbidity, hence the light can penetrate through and the algae can grow and subsequently use the nutrient in the wastewater. Can you go to the next slide? However, when we look into phosphate and ammonia, we find that the removal efficiency was very high at 60%. If you look into the graph, the graph in the middle, graph B, you can see that the leg and log phase is shorter in B, which is 60%. This is where we observe highest COD removal, and the fact that the leg phase is short is desirable because you don't want your water to sit for long, as opposed to if you look at the leg phase is long, which means it takes a long time for you to get the highest removal, nutrient removal. Can we go to the next slide, John? So that corresponded with, can you click again so that it can highlight where we get highest chlorophyll? If you can see it highlighted at 60, where we get highest chlorophyll, which correspond with the results that, because when the algae grows, it uses your ammonia phosphate and nitrate. So we get highest chlorophyll, which is the green color that the algae it produces as it grows. And we also get highest dry cell weight at 60 for all 4 farms that we work. So this means that FICO remediation works the same way for all 4 farms, that we observe similar trend in all 4 farms that we work with. So which means the water requires dilution of 60% for algae to work well. Can you go to the next slide? So these graphs are just a representation that shows that algae is actually growing. So this is the microscopic pictures which we took over the time of the experiment. You can see that these are the cells of the algae that we took over the time. But when we finished the experiment, we observed that there was like the chlorella species, which is a type of algae, was dominating. And it was not something that we were shocked, because chlorella species is a species that is very adaptive and it can adapt in different temperatures. So we realized that over time of treating, because we ran the experiment for 15 days, over time of the experiment, the chlorella started to take over. Next slide. Then we decided to go for field trials. When we were going for field trials, we chose to use the processors because when we look into the type of water that is in the slurry, it was not desirable for large scale because of the high solid content and also because of the turbidity. So we decided to go with the processor because normally the processor wastewater, it does not have as high solid content because the solid affect the light penetration and also the color of the water affect the light penetration. As much as the processors have white color, like the water is white color, but it's not as dark as the dairy farm. So we choose to do the field trials in the processor for that reason. Next slide. Okay, so we started this experiment, this large scale experiment at in June, where we place four ponds, pools, which were 3,000 liters, and you can see if you look into the first one is the seed, which means when you're growing, you need to have seed, which means the inoculum that you're going to inoculate. This is our algae that we have and we brought there. So we need to put it there as a seed so that we can inoculate other tanks. And also by putting our seed there, we sort of like want our seed to adapt to the environment so that when we start the experiment, when we start to inoculate the other pond, it can be a seed that is already adaptive to the environment, to the local environment. Next slide. So we continue with the same dilution, but now we find out that 60 and 50 are very close because the first experiment that we did, which was adaptation, where we were adapting the algae into the dairy wastewater, we started with dilution whereby we have 50 and 60 dairy wastewater in 100. And then we realized that no, 50 and 60 are too close. Let's make it 75. Also, by making it 75, we are also reducing the volume of fresh water that is going to be used to dilute. So we ran the experiment at 50-50 dilution, 70-25 dilution, and 100. And we also had our seed. Our seed is the one that we take and then inoculate the wastewater. So we inoculated with 10% of the seed in each pond. Next slide. So you can see from the top pond, as you see that that's how the natural color of the wastewater is. So it's sort of like whitish and have some fats. As it grows over time, it grows and becomes green, which is the chlorophyll color which I was talking about. So as it absorbs the light, and form the green color. So over time, after we finish the experiment, it changes into that green color, which shows that the algae is indeed growing without even going to the lab. So we could visualize it by looking at the color changes in the pond. However, when we were running, unlike in the lab, because in the lab, when we were running it in the lab, the conditions are controlled, your temperature is controlled. However, in the field, the temperature is not controlled. Everything is just like, we cannot control how temperature is. So what we observe there, when we start, if you look into the other graph with 3 points, when we start, we realize that the natural bacteria which are in dairy wastewater, they started to change the water into red, which those bacteria, we call them photosynthetic bacteria because they also absorb light. But what happened is that those bacteria actually help us because the COD were very high in processor wastewater. So it was so high for dairy to thrive in. However, because of those bacteria, which are normally known to coexist with lactic acid bacteria, the lactic acid bacteria, which is known to be used in dairy industry, they actually, as they grow, they use the CODs and lower it to levels where the algae was able to grow. Hence, over time, we saw that the algae was starting to change color from red to green, and it was growing because now the condition were conducive for algae to grow. Next slide. So before we run this experiment, we also, because before you run experiment, you need to establish the concentration of the wastewater that you're going to be working with, so that you know if you need to do dilution, if you need to do additional step to treat the wastewater. So what we did monitoring for 3 months. And when we were doing, we work with 2 processors, and we realized that I'm going to draw your attention to the red color, which is the wastewater that we took from the sprinklers in each processor. So in each processor, we find the water that we took from the sprinkler was above the limit, which also again tells you that a treatment that is required before the water actually goes to the environment. So we did the same thing. We did the monitoring for the secondary farm and for the second processor, and we also observed the similar trend that the contaminant were way too high as compared to the standard, which is prescribed by South African government. The next slide. So as we were running the experiment, we ran the experiment for 30 days, and we took sample every 4 days, and we analyzed for the contaminant for your COD, ammonia, and we find that the highest removal efficiency was observed at 70%, which means also because of the color of the processor wastewater, it requires some sort of dilution, but in this case, we were happy that the dilution require only 25% of fresh water, because we don't want to use a lot of fresh water. So we observed that at 70, when you diluted it with 25% of water and have 75% of the wastewater, you get the highest removal efficiency. For COD, yes, it's still high, it's still 1,000, but it is a removal when you compare to how it started, because the COD can go as high as 5,000 in dairy processors. So as much as it does not meet the standard after removal, but it is it's quite high considering where it started at. And we observed that at 100, the removal efficiency was not so high for both COD, ammonia. And the next slide, go to the next slide. If you look into the nitrate graph, you will see that there's fluctuation. Someone would ask why there's fluctuation. It's because when the bacteria within the water, they are resident, the natural bacteria of the wastewater, when they break down the ammonia, it breaks into nitrate. So hence you see fluctuation as it goes down and then it go up, because when they're breaking down the ammonia, there will be a spike in nitrate. And then because algae prefer to use nitrate, and that's why you see it going down. However, what we observe now in the reprocessor, we saw that the algae was not so good in using phosphate, because as you can see at the graph, there's not so much decline in phosphate. So we can conclude that the algae can use both CODs and also ammonia and nitrate. However, for some reason, the removal of phosphate was inhibited in this case. Next slide.
So during the cultivation, we also during the treatment of the dairy wastewater, we also see how the biomass algae is producing biomass. As Dr. Nokumisa was mentioning earlier, the red color of the cultures, photosynthetic bacteria is taking. So it is totally different when you grow algae for dairy wastewater treatment in the lab and the field trial. It was totally, because it's a control condition in the lab, and the field trials are natural. So you can't control on the photo period, light intensity, pH, temperature, and the weather also. So you can see in a first line graph in the left side, you saw the algae physiology, FEFM. It's a microalgae. FEFM represent the maximum photochemical efficiency of photosystem 2. It is crucial parameter used to accept photosynthetic activity and impact of environmental stresses. A higher FEFM is a 0.5. Is a, if FEFM is a higher than 0.5, it shows that your algae is growing perfectly without any stress. And if it's growing less than 0.5, it shows there is a stress. So you can see that all three dilution till 20 days in a first line graph is a below 0.2, means it's a growing in a stress condition, because it's totally is a field trial, open pond system. There is a different impact of the weather or the light intensity is what, but it doesn't mean it's not growing. It's growing, but it's in a stress condition during the all environmental factor. But after 20 days, you can see the peak is going up, because this is the condition the algae adapted. If you see the grow in the lab condition, it take only 7 days to go an adaptation period and pick up, go the FEFM value go above 0.5. But here it took longer. It took almost 20 days for adaptation period, and then it pick up the FEFM value. And the right side, you can see the bar graph, the value of the biomass also above 1 gram to, and it was almost more than 3 gram per liter in all condition. The maximum was in a 100% was showing, because most probably due to there was some solid content in was the dairy wastewater. It was also contributing in a biomass. Next slide, please.
Oh, okay. So after we run the experiment both in dairy processors and in dairy farm, what we find is that microalgae is capable of treating both dairy wastewater from dairy farms and dairy processors. And we also find that upon growing algae in this wastewater, you can get high dry weight cell and of algae, which can be used for various purposes. Although we did the studies, there's still more studies that needs to be done in order for us, like we have mentioned, it takes 20 days under stress. So there's still more studies to optimize it, to make sure that it takes lesser time to grow under this wastewater. So we managed to show that it can be done. Algae can grow and can reduce the contaminants in dairy wastewater. However, we still need to do more work to make sure that it's applicable to dairy wastewater. Next slide.
Dr. Lohi.
Okay. As we have discussed, we know that all the wastewater is coming there. But most importantly, one thing what we have observed is that the nutrients, which is apart from nitrogen and the phosphorus, there are still other nutrients which still left behind in the whole wastewater. Particularly, one is the lactose, and other nutrients like nitrogen and phosphorus still has present in the wastewater even after the treatments. And whatever the biomass we are getting through the algae or anything, we can still use it for some other derivatives like biogas, biohydrogen, and even like a single cell proteins. And most importantly, we need to reuse the water. Like being in a dairy industry, we know that we need the water. Like it's kind of a raw material, just like the milk. So without the water reuse, it will be difficult in a later stage for when we are thriving for the using all the water and all. So even for the lactose, John, can you go for the next slide, please? Yeah, when we are saying for the lactose and all in the wastewater, dairy wastewater, particularly, it almost like 5%, minimum 5% lactose still it will be there, and most of the bacteria derived from the dairy or milk only they can utilize the lactose, but algae cannot completely utilize it. So we are looking for the lactose, like as we've seen that whole wastewater which is present, like for one particular farm, minimum like a 10,000 per day wastewater they are generating. That means we still have a other part of the lactose which is present is almost like a 500 kgs of lactose, which we are actually releasing to the environment sometimes. Though those lactose we are actually focusing on how we can extract it for so that we can utilize it for further purposes. And again, for the water treatment, we should always focus on the reusing of the some part of the wastewater. Like since we are focusing more for the sustainable goals, like once the lactose is extracted, we will be using some other uh, strategies to extract the lactose. So whatever the water remaining will be having a potential to reuse it, other than the agriculture irrigation also. So that could be a better future aspect that we are looking for. These two are the one. Yeah, I think next. Next slide, please.
Thanks. Yeah, that's
Thank you, John.
thanks for all the presentation. I guess, you know, as just to sort of introduce the closing discussion is, you know, we've had a lot of conversations about FICA remediation and its feasibility. And I think the answer that we've come up with, does FICA remediation work? Yes, it does work. And it does work in the context of dairy farms. Is it feasible? Yes, but with caveats, is that it does require a bit more research. It also requires further adaptation and sort of investigating how it can be enabled more effectively, particularly in the context of dairy farming operations. So for us looking forward is, you know, one of the big issues with the dairy farms is the single slurry pond irrigating directly onto the pastures, and the potential issues associated with not meeting the thresholds as specified in the National Water Act. And perhaps one option to address both elements and also to enhance the potential for FICA remediation is to consider a three-pond system, such as a waste stabilization pond, which involves an aerobic pond, followed by a facultative pond, which is aerobic and aerobic, and a final polishing pond, which is aerobic specifically. So that's something that has certainly worked in other contexts, which I think we should be exploring. Can at the scale we've been operating, it's been effective. We now need to look at how we could potentially scale up these to larger scale operations. And I think, you know, one of the challenges with dairy farming is a lot of it's located, or certainly in KZN, in the Midlands, which is subject to very cold winters with reasonably short day lengths, which certainly had a notable impact on the performance of the system. But at the same time, and as Misa indicated, you know, you had this almost symbiotic relationship between the lactic acid bacteria and the microalgae that were sort of working in tandem to address the biological and the chemical oxygen demand. So can the system be adapted to become more effective? And in particular, as Dr. Faiz said, is to reduce the period or the residence time of the water to allow for more water to be processed within a given period of time. So I think those are the areas that we certainly would like to focus on going forward. But, you know, I think it's also critically important that we understand what farmers and processors really consider to be important. We know that we have to be good stewards of the environment. We also have to be, you know, enterprises need to be profitable. So it's about balancing these environmental and compliance requirements with profitability of the enterprises and sustainability in the long term. So, you know, it would be really nice to understand what stakeholders consider practical at a farm or at a processor scale. And, perhaps have some ideas around what are the opportunities and challenges to enhance the adoption of phycoremediation and waste stabilization ponds within the sector. Yeah, so I think those are our concluding sort of comments. And yeah, thanks for your attention. We appreciate it.
Okay, and now if anybody has any questions, you can use the raise hand function and I will unmute you. Colin, you can ask your question.
Thank you so much. Really fantastic presentation, and certainly having a look at those results, definitely corroborates in terms of being effective, especially on the COD. It's very interesting to see that. I think from a on farm, I'm thinking farming, but both farm scale and processor scale. One of the big challenges, and I think you alluded to that during the presentation, is obviously the holding time that would be required to enable the algae to do what they do. If you 20 days, to have 20 days to do the sort of bulk treatment, every 20 days inoculating. I think that's going to be quite a challenge. I'm not too sure how one gets around that at this stage, but we'll leave that up to you guys. But yeah, I think just having that bulk storage capability to be able to do mass inoculations, and then obviously providing sufficient time for the organisms to do their work, it's going to be difficult.
Thanks, Colin. Yes, absolutely. I mean, and perhaps our colleagues at DUT can talk to this. In closer to the coast, it's certainly the residence time is a lot lower due to the warmer and more stable temperatures associated with being near the coastline, and it is something that we'd have to look at. And I think it's a combination of two components: is pond configuration, and multiple ponds would certainly help to accelerate the process, and then adapting the microalgae to these, I guess, more challenging environmental conditions that you would find in your higher lying areas. So I guess those are the two important things to consider. But we've got to also remember that the small scale experiments that we're seeing at the moment are little plastic ponds. They're exposed to the environment. They're not in the ground. So again, there's already a much higher thermal variation, both between day and night and across seasons, because pretty much a larger proportion of the water is exposed to the environment. I don't know if our colleagues from the DUT have anything to add on that.
Yeah. So as I mentioned during my presentation, that this still needs work. We have shown that it can work, but the research all still need work, because although it took time, we can consider using multiple ponds, and also consider using the natural algae that grows in those conditions, which is already adapted to those conditions, which are points which we didn't go into, because when you use the algae which is already adapted, which comes from those conditions, it might be able to adapt faster than the algae which we took from the lab. This algae that we use in this experiment, we took from our lab, which is a very warm condition, and we took it to a area which have very cold conditions. So of course it's going to take time to adapt to those conditions. But what would have happened if we use the algae which we isolated from that side, which is colder region? We didn't go into depth to that. Hence, I mentioned that we still need to do work in terms of optimizing the system. These results only show that it can work, but we can still work on it to optimize it, to reduce time, and also figure the configuration that we can use that will reduce time in terms of how many ponds that we can use, how we can use it. Can we use them in parallel, or can we use just the configuration? So yeah, we still need to work on that. Because the time is a crucial factor. At the time we ran the experiment, it was very cold. It was like there would be snow at that time. So I think if we use the algae which is used into those conditions, it would have performed better than the one that we took from our lab.
Colin, I want to add one more thing. As I mentioned, in our lab, is a condition was control. So efficiency was very high in compared to our field trial. Earlier, Dr. Nukumesa was saying this that time when you were growing algae in the site on field trial, this was frozen, its temperature was very low. And the algae is a photosynthetic organism. It's needed a temperature above 20 degree to do photosynthetic. When they do photosynthetic, so the photosynthesis, they will take all the nutrient or reduce the COD. If you do the techno-economic analysis, how this technology is feasible, you have to consider all how it will go. If you see this technology is a zero waste producing. If we are using algae phycoremediation to treat dairy wastewater, we are not using any type of chemical to treat wastewater. We are using sunlight, and base of thing, we are also sequestrating the CO2 from the environment. And the dairy industry can talk to the government for the carbon credit. For 1 ton biomass you are producing, we are 2 tons of CO2 from environment you are sequestrating. So you see you are closing the loop. You are not using any nutrient. You are not adding any light from any sources. You are not just what about the nutrient inside the wastewater. Algae is taking for growth, and you use the biomass. This biomass produce, you can use a fertilizer, and the water after harvesting you can use for the irrigation, and you can reuse for the dilution. Here we are using for the tap water for diluting. We can reuse for the dilution further. So multiple pond will be a most suitable a strategy to go further. And when you're using multiple pond, so that time you no need to have a more like a seed culture. We have to add 1 time of seed culture. This pond will go the other pond. So 1 time you have to inoculate. And when your algae is adapted to that condition, there is no any situation that your pond will crash and I think anything will happen. So that will be overall, if you do techno-economic analysis, the thing will come in a positive. Hope I think I answered the question.
Thanks very much. Thank you.
Does anybody else have a question? Mark, you can unmute yourself and speak.
Thank you. Yes, a very interesting talk. I just want to know, have you considered looking at perhaps water plants as an alternative to algae? I'm specifically thinking of the invasive plants. I know obviously there's environmental issues, but plants like hyacinth or Kariba weed, which also obviously grow at very rapid rates, or are those not viable options to look at? Thank you.
Some student is doing on the lab scale, but we have a facility in Etukeni municipality. There is a plant in a Manjimtoti wastewater treatment plant. We have 300,000 liter raceway pond where we are using this phycoremediation technology for polishing purposes for This is a domestic wastewater treatment plant, and inside the domestic wastewater treatment plant, we have a 300,000 liter reservoir pond using this technology to polish the wastewater. But what you were saying, some student, I think, is a undergraduate student doing on the lab scale, the early, like you can say, preliminary experiment they're doing. But in a wastewater, using for algae, domestic wastewater, industrial wastewater, we have a well established on that research. So this is the first time we are moving to from domestic wastewater and industrial wastewater to the dairy wastewater, and we found, like, it's an interesting result.
Yeah, I think just to add, I think the scale that would be needed for something such as water hyacinth would be a very shallow pond with a very large surface area, which could impact on the feasibility, I think, is the one point. And I guess it would also be quite challenging to get environmental affairs to approve the use of a serious invader to treat wastewater, particularly given some of the challenges we're experiencing on some of our dams. Although there is a lot of interesting work being done at scale on these dams to look if they can sort of remove nutrients and recycle biomass at scale. But I think I'm not sure that it would be appropriate for a farm scale or a processor scale kind of system. Yeah, thanks.
Any other questions? Yeah, doesn't look like anybody else has questions.
Are there, I mean, perhaps just a closing question, are there any sort of major red flags that anyone can see that would sort of prevent this from being taken forward, or anything that should be, that we haven't considered in sort of trying to really get to the bottom of the feasibility elements of research like this?
If I may come in here, I would say that the practicality, and I think if you've actually mentioned it in your talks, is having a multi-dam system where your first dam is your sedimentation dam. Because from what I've seen on farm visits, that first slurry dam frequently has a very thick, hard cake of cover of feces that will prevent the growth of algae. But the subsequent dams, where the water, sort of the overflow dams from there, the water is a lot clearer, and I think the algae will grow really well in those. So any of the farms that have a multiple dam system for their wastewater, I think that'll be, I can't see any problem with implementing this.
Okay, great. Thanks. That's very, very helpful.
That's, John, this is very helpful. Like, we no need to do further dilution.
Yeah.
So it will be clear. So you can save the water also.
Yeah.
Alan, you can go ahead.
Shane, thank you. Yeah, just also to add on to what Mark said there, I think in your lab analysis, you obviously had to do those dilutions to see the efficiency of the algae improve. And yeah, I think if one could come up with an optimum number of ponds, obviously depending on the amount of wastewater that's being produced on the dairy farm, have a point along a string of ponds where you feel then it's optimal to introduce the algae. I certainly think that's something that could be done, and then also perhaps some sort of pre-treatment, coming off the farm itself, whether it be the use of a screen or a manure press or something, where you removing the bulk solids from the effluent stream before discharge into the initial pond. Could potentially just help reduce load on the effluent before one looks at introducing the algae.
Cool. Thanks, Mark and Connor. I think both those inputs are very helpful. Thank you.
Wow, I think we are going to end it off This was really interesting. Thank you so much to our 4 presenters. Well presented, wonderful information. Thank you so much. You've really added value to our industry, and we greatly appreciate it. All the best for your future research. We will get you back to come and speak to us again. But thank you so much. We greatly appreciate you. And then I just want to mention our next webinar will be on the 28th of May, and Dr. Mark Charms will be discussing brucellosis. So please have a look at our social media, register for the webinar, and then we'll see you back on the 28th. And then if everyone is happy and there's no more information or questions, I'm going to wish you a wonderful afternoon. God bless.
Thanks, everyone.